Hello everyone, welcome to Gebze Technical University Department of Bioengineering webinar series. Our guest for today's webinar is Research Assistant Dr. Orkun PİNAR from Marmara University, and the webinar title is Valorization of Waste into Value-Added Products Through Bioprocesses. Research Assistant Dr. Onur PİNAR received his bachelor's degree from Ege University, Department of Bioengineering in 2006. He completed his master's degree in 2009 and his PhD degree in 2017 in Istanbul Technical University, Department of Molecular Biology, Genetics and Biotechnology. In 2017, he carried out a 3-month postdoctoral research at Arizona State University. Since 2011, he works as a research assistant in Marmara University. Hocam, you can start.
Thank you very much. I'm sharing my presentation. Good afternoon. So first of all, I'm really appreciated for being here for your kind invite. So my name is Dr. Orkun PİNAR from Marmara University, and today I'm going to mention about the valorization of waste into value-added products through the bioprocesses. So you can see some pictures in my cover slide. So I'm going to mention about this kind of products through different kind of systems. First of all, I want to mention a bit as a summary for the bioprocessing because it's an essential part of many food, biochemical, pharmaceutical industries, and not only for the cheap materials like the industrial alcohols, organic solvents, but also for the expensive biochemicals, including antibiotics, therapeutic proteins, and vaccines. That bioprocesses are very important. So also in that systems, we need a really bioreactor so of course in an industrial scale, and also under the optimum conditions, gas, I mean the air, oxygen, nitrogen, and the carbon dioxide, according to which kind of organism are you working with. These flow rates are very important. Temperature, pH, dissolved oxygen level, agitation speed, and these conditions are very crucial for a rapid reproduction of the microorganisms or cells in that bioprocesses in the bioreactors. Also, when we look at that, if we go further for the title of my presentation, I must say that there is a huge potential of waste. Sometimes it's a bit strange because if we are talking about the potential of waste, but we must say that it's expected to grow to 3.4 billion tons per year in 2050. For now, we're dealing with among 2.2 billion tons of global waste, and the most abundant one in that food waste is the food waste, and it's almost 1 million tons they are generated in 2019. And also the same in Turkey, there are 12 billion tons of fruits and vegetable waste, and it's almost worth 4 billion euros, which is generated annually. And while we're talking about that potential of waste, today I'm gonna put an emphasis on the cereals, like the fruits, vegetables, and nuts, husks, skin, peels, and shells, which are very important because they are full of lignin, cellulose, and hemicellulose, and especially cellulose and hemicellulose part are very, very crucial for sustainable bioprocesses. Also, if we talk about the value-added products, because the materials including the technical enzymes, biopolymers, bioplastics, organic acids, and the alcohols are industrially important, and with the conversion of that waste into that value-added products become very, very crucial in terms of the sustainability. And when we consider the numbers, the amount of waste, that increasing amount of waste that I mentioned before. Also, using recombinant and the native microorganisms can be used through the bioprocesses with a green chemistry approach for creating a more sustainable environment and the society. So therefore, the scientists, academicians, and companies put emphasis on the solutions regarding the reduction of waste and use that waste for production of value-added products. So when we consider that value-added products, actually, with that convergence, enzymes are very, very important. And so, first of all, I want to mention about an enzyme which is called Laccase, because it has a full of different properties. So first I came across with that kind of enzyme in my PhD, and then I saw that it's a very promising enzyme also for a lot of different conversions of waste into the first of all fermentable sugars, and that fermentable sugars can be used for further bioprocesses. So first of all, I want to mention about a Laccase from Coriolopsis polyzona and recombinant production of it. And step by step, I'm going to mention about different kind of cases in terms of that topic. So Laccase is an oxidoreductase, and they are a superfamily of the multi-copper oxidases. And they have different biotechnological processes. They take part, like the identification of the lignocellulosic biomass, and bioprocessing, the processing of the beverages, some medical diagnostic tools, especially bioremediation, because they can clean up pesticides and herbicides, and for the detoxification of industrial effluents. And sometimes that industrial effluents can be a good candidate for, again, getting value-added products with that, and baking, and also they can take part as an ingredient in the cosmetics and in biosensors. And for the industry, we need large amount and highly active enzymes, so there's a huge demand of that, especially heterologous expression of the clone laccases are very important, because with this modern techniques of the recombinant DNA, we can get desired modifications, low cost, reduced timing, and lower cultivation disadvantage. We can lower the cultivation disadvantages. So the cloning of the laccase genes and heterologous expression can be performed in bacteria, yeast, filamentous fungi, and the plants for better laccase production. Also, precisely for this work that I mentioned in the headline, so the Coriolopsis polyzona is a white rot fungus and is a promising industrial enzyme producer. It has several enzymes, and laccase is one of them. So in this work, it's aimed to isolate and characterize the full-length cDNA encoding laccase from Coriolopsis polyzona, and then heterologous expression in the Pichia pastoris, which is a metallotrophic yeast. And at the end of the work, there's a biochemical characterization of that recombinant laccase, which is very important to determine the properties of that enzyme for the further industrial application. So also, this is the first work cloning a laccase gene from the Coriolopsis polyzona. So if I show you the how we can get it, so this is, you can see that in here, there's the PDA agar in here. So before and after, you can see the how it is gathered on that plate, and after the vegetative cell growth, and now we have to separate the vegetative cells and the fungal spores, and after cleaning fungal spores with a sodium chloride solution, and we can get a very clear white spores, and we can use that spore for the inoculation of the production expression medium. And at 10.108 we have a culture. So if you just determine the laccase activity from that one, as we did in here, so we have the laccase activity as a crude laccase from the C. polyzona cultures. So, and then we use the pellets that we get from that culture for the RNA isolation in order to get the full-length sequence of a laccase gene from Coriolopsis polyzona. For this purpose, we first make a cDNA synthesis, and also we make a PCR with the degenerated primers. Why this is important? Because laccases are copper oxidases, so that's why they have some copper binding region, and these regions are very conserved. So we use this advantage in order to get almost the middle of that cDNA from here and to here with the N primer and the C primer. And then after that amplification, we'll make a ligation and the transformation to the E. coli, and with the conformation with the plasmid isolation and the restriction enzyme digestion, and then just next to the sequencing, we saw that we come across with at least 3 different laccase genes in that organism, and also in the literature, and it could be up to like 15 and 70 different laccase genes in that matter. So that's why when we look at that library for the construction of cDNA library, we saw that the most frequently detected sequence is a CPLCC1, and we go further with that cDNA. As I mentioned in the beginning, now we have the middle of that gene, and with some different reactions, we call it race, rapid amplification of cDNA ends, we should provide the 5' and the 3' of that gene. So at the end of that reactions, we could get a full-length CPLCC1 cDNA PCR product. So why this is important? Now we will have ability to express it. So after the ligation and the transformation to E. coli, and after that conformation with the gels, and we get the sequencing, and we get the whole sequence and full-length CPLCC1 cDNA. Now we're just at the gate of expression. So there is a vector in here, you can see in here, PPZ, and we use that shuttle vector for the expression that full-length CPLCC1 cDNA in the Pichia pastoris. And now you can see that place, which is supplemented with the substrate. So if there's an extracellular production of the Laccase, you can see that zone. So you can see a lot of green zones. So it means that your transformers are producing Laccase enzyme. And after that, we just choose the best producers. And then after one producer, you can see it, and at the beginning, it is 33. So we made an optimization of the production, and we continue with the partial purification of that Laccase enzyme. And at the end of the day, we just continue with the biochemical characterization in order to see the properties of that enzyme. So for the optimization part, as I mentioned, we just use BMGY and BMMY mediums for which is specific for the Pichia pastoris. And also we mentioned about the optimization of the methanol concentration, because it is so important. Methanol is not only a carbon source for the Pichia pastoris, but also important for the induction of the alcohol oxidase 1 promoter, which is important for in that system, important for the expression of the Laccase gene. Also, copper is important, as I mentioned in the beginning. Laccase is a copper oxidase, so the copper is very, very crucial. So also we make a copper concentration optimization using the copper sulfate. Also, interestingly, we made another optimization based on the L-alanine, because you know that with the during of the production, and we're talking about the 8 days in here. So there's a lot of byproduct production step by step, and this byproducts makes the environment's pH into the very acidic conditions. So in order to get rid of it, to make a balance, so we use an alanine in here, so that organism can consume the alanine and just produce some ammonia. That ammonia just make a balance in the environment for the production, because that lower pH become very, very harmful for the period of that production. Also, we tried for the growth temperature optimization, and from the like the 23 and the 28, and at the end of the optimization work, and we have 9.8-fold increase in the Laccase activity. And just we have using the same medium, but we changed the concentrations, 1.1% methanol, 0.8 millimolar copper sulfate, 0.6 percentage L-alanine, 28 centigrade, 250 RPM, and for 8 days. Also, after that, as I mentioned in my flow chart, I mentioned about that we go for the biochemical characterization, and we just determine the effect of the pH and the temperature on the Laccase activities. Also, we just investigate the different substrate and inhibitors on the Laccase activities. And finally, we just get the catalytic properties of the enzyme, which is very important for its reaction. So when we look at that, after we get some optimized conditions, as I mentioned, for the methanol, copper sulfate, L-alanine, and temperature. So also with the temperature work in the biochemical characterization, we saw that the optimum temperature for this Laccase enzyme is 70, and the optimum pH is 3. So also the thermal stability and the kinetic parameters of that CPLCC1 Laccase become promising, and this is a high redox potential lacase, and this makes that CPLC1 lacase a good candidate for the industrial applications. And also, after that, I just met with the Laccase enzyme, so I saw that there are lots of work with the Laccase enzyme can be done, and in a parallel way, just we start another project, and also we just try to get the for the next level. We know that we have to scale it up because it was at a lab system, is a lab level production, and also with different kind of protein engineering approaches, we know that we can increase its properties at the high level for different industrial applications. As I mentioned, after we saw that the lackases are very important, and in a parallel way, we just start another project, and now we want to use that enzyme for the especially get the fermentable sugars from abundant lignocellulosic waste. So when we look at that, we know that lignocellulosic biomass is the most abundant renewable carbon source on the earth. So that's why there are lots of attraction to them for the production of the value-added chemicals, because obtaining fermentable sugars from this abundant biomass is critical. And if we can do that, we can convert that waste into the value-added products because they have lignin, cellulose, and hemicellulose complex. So if we use lacase, we can get rid of the lignin, so we can increase the penetration of the cellulase enzyme or hemicellulases in order to get the sugar just hidden in that lignocellulosic biomass. Fermentable sugars are important, and there are different methods. So using like the alkaline acids or different kind of more harsh conditions. But as a result of that one, the byproducts as a result of that procedures become very harmful for the microorganisms. So that's why, and instead of that one, enzymatic hydrolysis could be a good candidate. So that's why in that project we just use a cellulase and the lacase as a solely and together in order to make a multi-enzymatic systems to get that fermentable sugars. Also, the pre-treatment steps, as I mentioned, is important because if you can just provide an efficient hydrolysis and you can just increase the economic feasibility of the fermentable sugar production. So again, instead of the conventional method, as I mentioned, as a harsh methods, so we can use the enzymatic hydrolysis, can be an advantage for the reducing of capital investment and improve the overall economic feasibility, which is very important for the bioprocesses. So this work is first evaluation of the effect of lacase on the enzymatic hydrolysis of hazelnut husk, which is one of the most abundant lignocellulosic agricultural residues which are generated in the Turkey. And as a result of that, we just try to get fermentable sugars for the further bioprocesses. So first of all, we use some commercial enzymes, and their temperature and pH were kept constant because the 45 centigrade and the pH 4.5 is optimal and stable conditions for that enzymes. We tried different amount of hazelnut husk from 4 to 80 grams per liter, and with the different kind of enzyme loadings for the hydrolysis. So, and then while we're using the cellulose, we use the volumetric, the equivalent lacase to the reaction medium in order to increase that enzymatic hydrolysis as a co-enzymatic hydrolysis. So the sugar concentration that we get from them expressed at a gram per liter and in that total reaction mixture. So when we look at that, there are different kind of loadings. So from the 4 gram per liter husk to the 8 gram per liter husk, and we saw that, as I mentioned, we use the volumetric equivalent lacases, and in the first graph you can see that this reducing sugar concentration made by only the cellulose, and in the second one it is cellulose plus the lacase for getting that fermentable sugars. And in here, in the first one, we get 1.32 gram per liter fermentable sugars after using the cellulose as alone. So of course, when we look at that, when we increase that from the 40 to the 80, we cause a slight decrease in reducing sugars. But also when we look at the Laccase and the cellulose treatment as a simultaneous hydrolysis, we get almost the same, a bit slightly decreased version of the sugar concentration. But there is another thing in here because it seems like the cellulase is giving a lot of, not a lot of, a more sugar content when after the hydrolyze. But when we see that, when we make a HPLC analysis with the Zorbex for the sugars, we saw that actually the glucose concentration in that reducing sugar is very low when we do that hydrolysis with only the cellulase. But if we do that cellulase and lactase simultaneous hydrolysis of that hazelnut husk, we saw that almost 50% of the reducing sugar just belongs to the glucose. So actually we know that only with the cellulose hydrolysis we get less than 10 of the final reducing sugar concentration in that hydrolysis. So we saw that coenzymatic hydrolysis of hazelnut husk by using the lactase and the cellulose simultaneously without the separate pretreatment processes that I mentioned, we get a glucose content of final reducing sugar concentration, as I mentioned, over the 50%. So also not only getting the fermentable sugars, but also we use that hazelnut husk as a substrate for production of that lactase and cellulose enzyme. So we use Picnoporus sanguineus, which is a white rot fungus again. So with different fermentation, in the fermentation medium, we just load different hazelnut husk proportions, and also we supplement that medium with the yeast extract and the malt extract. And after the micellar suspension was added to the fermentation medium in the flask, so after the 10 days, we harvested the cells, and then we get the supernatant for the enzyme activities. And after the ultrafiltration, we get a more concentrated supernatant, and we measure the activities of the cellulase and the lactase enzymes. So when we look at that, the cellulase, because while you are determining the activity of the cellulase, you should check the beta-glucosidase, CMCase, and the filter paper for the FPase, filter paperase activity. So when we look at that, we couldn't detect any filter paperase activity, but we saw that the CMCase activity is 21-fold higher than the beta-glucosidase activity, almost to 4,050 units per liter, and also we get 2,240 lactase activity at the end of the 10 with the 12 gram per liter heart concentration. So we saw that the Picnoporus sanguineus produce lactase and the cellulose mixture incapable of degrading cellulose and lignin found in that hazelnut husk, because we must say that the over 50% of the that hazelnut husk is from the holocellulose, cellulose, and the hemicellulose, so that's why it's a very important feedstock. So depending on the fermentation period, enzyme mixtures containing different percentages of lactase, CMCase, and the beta-glucosidase can be obtained from that work. When we look at overall, so when you look at that again with the only cellulose usage and the cellulose plus lactase usage in that manner, we can reach about 50% of the reducing sugar as a glucose. So the addition of the lactase just resulted in possible identification of the lignocellulosic material. Why this is important? Because after that identification, it has just become more penetrable for the cellulases, so the glucose content is increasing step by step. Also, we saw that the hydrolysis of hazelnut husk by enzyme mixtures without using any harsh pretreatment process is possible. Also, with the Picnoporus sanguineus, a white rot fungus, as I mentioned, can use that hazelnut husk as a carbon source, so it becomes a promising feedstock, and also it's a waste because people in Turkey just putting it, just burning in the ovens, or they are just putting it just under the animals. So instead of that, we just use that valuable waste actually into a value-added biochemical products in terms of the fermentable sugars and enzymes. So the results obtained in this work help us to develop bioprocesses to generate value-added products and pave the way towards the bioprocessing. So when we get that information, so the data that we get from the actually from this project and also from the literature, we just want to make a bioprocess design paper based on that information. So after that, after that we saw the potential of that enzymes. We just published an paper called Bioethanol Production from Turkey Hazelnut Process Design and Economic Evaluation. Just with the idea that with the hydrolysis and the fermentable sugars, now in this paper we use that fermentable sugars for further production of the bioethanol. So the potential of that enzymes, we just publish the data coming from our wet lab and also the literature information. So that's why we just designed a synthetic bioprocess of an innovative and unprecedented bioprocess plant converting that Turkish hazelnut husk into the lignocellulosic ethanol. And while we're doing that, we also put an emphasis on the economic evaluation. And preliminary economic evaluations are performed on several scenarios in here. I'm going to mention about the 5 actually different cases with different properties step by step very quickly. So that changing that properties, changing that parameters for the bioprocess, just changing the inputs, and sometimes we change the plant capacity, evaporation unit capacity, the enzyme price, nitrogen source price, and these are all simulated through that bioprocess design work. So this is the bioprocess. Actually, you can see it on the right, which is made by the SuperPro Designer, which is a computer tool to model, evaluate, and optimize the batch and continuous processes. And here we just use that tool for not only the calculation of the bioprocess design parameters, but also we just use it for the important economic metrics. And these are, as you know, that indicators of feasibility and profitability of the proposed process. So when you look at that figure, you can see that there are 4 different stages. And the first one is the pre-treatment. And when you look at that, the dried hazelnuts is coming from here. And step by step, there are some washing steps, there are grinding steps, and there is storage. And after that, we saw that after the drying and they are grinding, and we have small particles of that hazelnut husk, which is necessary for the enzymatic hydrolysis. And then we use that husk for the hydrolysis in that tank for the enzymatic hydrolysis. There is an enzyme mixture full of cellulases and the laccases in here. And after that, we use an evaporation in order to get rid of the excess water in order to get a condensed sugar. And so there's a flow splitting in here. We just put it one to the seed fermenter, one to the main fermenter for the production of the ethanol. So when we look at the fermentation part, seed fermenter is for the pre-culture of that ethanol fermentation. So at the end of the day, we get a fermentation product, and this is full of the cells and the bioethanol. So that fermentation product in the 4th stage as a downstream operations. After the microfiltration, there's a waste over here, and there's a product in here because that waste should be full of cells, because in order to get the ethanol, you have to get rid of cells. So there's a storage of the ethanol in here, and with the heating and the double distillation in here, at the end of the day, we have almost 95.5% bioethanol, which is made by the using the hazelnut husk as a feedstock in here. So you can see an overall mass balance in here. So it's the batch system. So for each batch, there is over 500 metric ton hazelnut husk coming in here, and precisely 542.7. And at the end of that bioprocess, we get 131 metric ton per batch ethanol. So So you can see also all the equipments, all the unit in here, and we use 4 parallel fermenter in here for getting this high ethanol from that process. And so, as I mentioned, not only the bioprocess parameters, but also the economic evaluation metrics are important. So for this process, the unit selling price for the ethanol is 1.5 US dollar per kilogram. And we just start with the hazelnut harvest because it is almost, it's changeable to the year to year, but we start with a 180,000 and then we just go up to the 360,000 metric ton per year. And we change the evaporation percentages from 25% to the 75%. Also, first of all, we just set the peptone and mix prices, enzyme mix prices, like the $1 per kilogram and $2 per kilogram, but also we just change it to the different cases. And because that enzyme mix price can be changeable within the 1.5 to 5 US dollar per kilogram, and also 5 different economic metrics, unit cost and return on investment, because this is so important. This is important because it is ratio to the investment in here. Also, unit cost is important because the operating cost per the rate of the annual production. Net present value is one of the most important, maybe the parameters in here. It is simply the all positive cash flows minus the all cash outflows. So that's why it's giving us a good parameter for the feasibility of that process. Also, internal rate of return is also, it is a true rate of return, and for local consideration, it is very important because it consists of the discounted cash flow and also the payback time. Of course, you know that in the biotechnology, so the payback time is, if it is just before the 10 years, so it's a good parameter for that. So that's why payback time is so important for the make that capital investment for that process, yes or no. So after that, as I mentioned, there is a base case, as I mentioned. So, and then we just try 4 different case study. First of all, this is changing the plant capacity. We just upgrade it to the 360,000 metric ton per year from the 180,000. And then, so when you look at that, especially in here, the unit cost, NPV, net present value, and also the payback time are the most important parameters, I think. And in here, when we increase that capacity, there is an overall decrease in the unit cost and the payback times, as you see in here, with the increasing capacity and MVP in here, and ROI and the IRR, all that increase. And this is important because if the NPV is very high, it has become a feasible. If it is positive, so it become a feasible process. Another one is changing the evaporation percentage, because after the hydrolysis of the hazelnut sucks into the fermentable sugars, there are excessive water in here. So that's why in order to use them as you want, so we have to get rid of the excess water. But if you do it so much, because it become infeasible, because sugar can be denatured, and also there are some solubility issues. So that's why the possible maximum possible level of the evaporation is 75, as we found from that design. Again, you can see that when it is increasing in the evaporation percentage, the unit cost is decreasing, MVP is increasing, and also payback time is decreasing. So that's why it's giving a positive property to that design. Also, the third one is the nitrogen source, because it is used so much, it has a significant portion. So that's why actually when we're increasing the nitrogen source, as you see here, the unit cost is increasing, but the MVP is decreasing. So that's why we must say that when we look at that 0 at the MVP, this is the critical point. Approximately after the $3 per kilogram, it becomes infeasible. So that's why the nitrogen source price above 3 US dollars per kilogram would make that process infeasible, and also that the negative values of the MVP just make that process infeasible. In the case study 4, if we change the price of the enzyme mix, and when we look at that, of course, in order to get the fermentable sugar, enzyme mix importance is inevitable. So arguably the most sophisticated raw material required for that process maybe in the beginning is the enzyme mix. So when you look at that, actually, you can see that there is no going to the under the zero or the MVP. Unit cost is again increasing, but it is not very effective. So you can see that unit cost is not very increasing in here. So that's why sometimes the issue could be for the changing price of the enzyme is based on the technological considerations. So what can you do? You can just search for the availability in-house technology, and sometimes time to market issues can be problem for decision at that point. But remember that in the previous project, we can actually get the enzymes using that technology, so we can use an in-house production for our preference for that getting the fermentable sugars. So when we look at overly, so we can see that base case study in here. So the other case studies take part in here. So yes, maybe for the case study 1, increasing the capacity of that plant is NPV is very increased, but we must say that actually the harvest of the hazelnut husk can be very, very changeable. Also, when we look at the pictures and the graphics taken from that case study economical metrics, we saw that after the 320,000 metric ton per year, it becomes some fluctuations. So that's why at the end of the day, and this is the highest capacity for that case study. So, but when we look at that, you can see that the payback time is very important, and it is very, very, all is under the 8. When you look at that, it's very similar to the base case study, and some of them are actually 3, and the 4 is better than in terms of the payback time when we consider the case studies for that production of the bioethanol. And when we look at the case studies and some uncertainty analysis which are not shown here, but the production of the second generation bioethanol from the hazelnuts is very important, can give us an economical potential. Also, some social and environmental advantages, because this abundant waste will become some value-added products and some fuels, actually, bioethanol, and they can be used in different kind of area. Also, not only for that fermentable sugars actually getting for the bioethanol, we also work with different kind of biomaterials. So one of them is actually biocellulose, bacterial cellulose. So another version of the valorization of the waste into a value-added product. We had a paper about that, the valorization of pea pods, celery root peel, and the mixed vegetable peel as a feedstock for biocellulose production from Comagotebacter hanseníi. So remember that while we're talking about the food waste, I said that I'm really interested in the peels. So this peels is coming from kitchen and can be a good candidate with their sugar for the production of the, not only, for example, enzymes, but also for the biopolymers. So bacterial cellulose is one of them. It's an excellent biopolymer, has a lot of has higher degree of polymerization, crystallinity index, high mechanical strength, and high water holding capacity. This is so important for the adaptation to different area. Also the biological adaptability, and then the plant cellulose, because bacterial cellulose is pure. So if you just start with the plant cellulose, it has lignin and the other parts should be discarded to get that cellulose, and there is always a level of purity problem for the plant cellulose. Also, the bacterial cellulose is GRAS. It means that generally recognized as safe by the FDA. So that's why it is important and can be used for many industrial area. And species belonging to Comagotebacter is formerly Glyconacetobacter. can produce a good bacterial cellulose with a high yield and the pre-give. And when we look at its market, it will reach a 5 million US dollar by the 2023. So that's why people just trying to find some scientific solutions to produce that BC. The bacterial cellulose is as a large scale and more eco-friendly bioprocesses like using agricultural byproducts and food industry waste. So at that moment, the objective of this work is to evaluate the vegetable waste as cheap carbon sources and use them for the bacterial cellulose production. And in here, particularly pea pod, celery root peel, and the mixed vegetable peel were evaluated to produce bacterial cellulose. And maybe their most commonly properties, they are not edible, they are just waste. So that's why we use that one to produce the bacterial cellulose. So the physical and the mechanical properties are very important for the bacterial cellulose. So that's why we also investigate them in order to make a comparison with the like the commercial media in order to see the effect of that for the rigidness and the other properties of that bacterial cellulose. So for our knowledge, and there is no work available in the literature that use the celery root peel and pea pod actually for the feedstock as a bacterial cellulose production. And so for this purpose, first we just get this waste and mix with the distilled water at the 1:1 ratio. So and then we shred it down with a blender to increase the hydrolysis efficiency to increase the surface area and it become more obtain the fermentable sugars in here. So we use two different methods, dilute acid hydrolysis and with the hydrochloric acid, and also together with the high pressure, like 15 minutes high temperature and in the autoclave. And also in other method, we just use high pressure and temperature for a longer time, like 1 hour in 121 centigrades, and then we filter them. And after that, we get a solution of sugar coming from that waste. And when we look at that, you can see in here, we get the maximum total sugar from the mixed vegetable peel. There are different conditions for that production. For the pre-culture conditions, we use the YPM medium, yeast extract, peptone, and mannitol for 28 centigrades for the 48 hours. And also we just use some Erlenmeyers with a 10% working volume for the BC production. And then we use the sole hydrolyzates. And also we use hydrolyzates with the yeast extract and the peptone, which is similar like the YPM medium, but instead of the mannitol, we use that waste solutions. So for the control, that's why we use an YPM medium for the bacterial cellulose production. And after the production, these pellicles just separated from the production media and incubated with the sodium hydroxide and for eliminate the bacterial cells. And then we wash them again and again, more than one with the deionized water, and that pH become neutral to remove the sodium hydroxide. And then we dry them, if necessary, at the room temperature. How it's seen, when we look at that, you can see that Comagotobacter hansen cells on the left, and the bacterial cellulose generation is happening here. Maybe one of the most advantageous thing about that production, and you can make it in a static condition. So that's why you don't need a shake. You don't need anything, something like this. And also with the larger area, you can get a bigger kind of cellulose production at this moment. So also on the right top, you can see a bigger one, which is made with a bigger static conditions as a result of that bacterial cellulose production. So when we consider that, we just try different mediums with different proportions of the carbon sources coming from waste. So first one is the control, which is based on the mannitol, but again, the other medium you can see in the table on the right, the celery root peel, pea pod, and the mixed vegetable peel also used for that manner. But when we look at that, we saw that almost 186 gram per liter bacterial cellulose produced based on that waste. So if we compare the first one, which is a control with the highest pellicles that we get as a bacterial cellulose, is there maybe the control is higher than in the weight when you compare the waste one. But if we consider the price of the mannitol, it's a very expensive carbon source that is become very cost effective to get that bacterial cellulose as a valorization of waste into that bacterial cellulose. Also, after we get it, we have some characterization. For the FTR, you know that FTR is for the having the has a characterization for the molecular structure. So when you look at that on the right, and there is a characteristic peaks, because if you just compare, the first one is the control, other ones coming from the bacterial cells, which is produced by the waste. And so this stretching and bending are very, very critical. And this is like an ruler for understanding the characteristic of your bacterial cellulose. So if you compare them, you can see that the peaks coming from the FDR is very close to each other. So these are characteristic peaks that should be provided in that bacterial cellulose. So also the thermal properties of the waste-based bacterial cellulose from the conjugated bacteria, we just identify them because it is very critical to make the identification of polymer characteristic. And also according to the application range, these characteristics become very crucial. So there are two actually different approaches, like differential scanning calorimetry. We just get the glass transition, crystallization peak, and also melting point. And when we compare it with the waste and the control, we saw that the glass transition is very important, and this is lower than the room temperature. The material become used as an elastomer. So when you look at that, and in here, in here, one is 10 and one is 3. So this one is the based on the lower than when you compare the control with the waste-based bacterial cellulose. It is lower than that, so it become more become potential to become elastomer. So also when you look at the TM values in here, so in here the control is 100 and almost 162, and for the waste 126. So this is also so important because if that has a lower TM value, the biodegradability is higher. So that's why we can say that the bacterial cellulose producing EFM medium is, when we look at that, lower than the waste. So waste has a more biodegradability. Also, the thermogravimetric analysis is important because it's really giving the thermal behavior of that polymer. So actually, at 330 and 340 centigrade, that both of them were the control and the waste bacterial cellulose showed a significant weight loss due to the decomposition, of course. But we must say that the thermal stability of the waste-based bacterial cellulose is better than the when we look at the other waste-based bacterial cellulose, which is given in the literature. For example, a BC membrane produced from a rotten mango is a good example for that. Also, we make a SEM analysis. You can see it on the top for the control, and on the bottom is the waste-based. But we must say that the waste-based bacterial cellulose has a thinner and thinner fiber structure than the control. So that's why the SEM analysis seems like that one in the characterization. Also, water uptake is very important because we can use that bacterial cellulose for different applications. So for that purpose, we just try the oven dried and the lyophilized bacterial cellulose for make a comparison. So oven dried bacterial cellulose membrane have a higher water uptake capacity when it is compared to lyophilized, because lyophilization can sometimes change the structure of the fibrils. So that's why it can be a problem in that situation. Also, antibacterial activity is important, there are two different work that we've done for that one. First one is to understand the commercial antibiotics, which is impregnated to the bacterial cellulose, and we just search for the E. coli growth with the effect of them. So also in the B in here, so we have a plant-derived agents which is impregnated to the bacterial cellulose, but when you look at that, they grow so much, so that's why the plant-derived agents are not sufficient for the inhibition of the microbial proliferation. But when you look at that, the control and the antibiotics in here, we saw that actually bacterial cellulose do not have antibacterial activity by itself, but it has a lot of capacity to impregnate with different kind of molecules into it. So that's why we see the effect on the left in the growth of the E. coli. Also on a plate, so you can see that on the A rifampicin and in the B the gentamicin. So that results show that that bacterial cellulose from the Commigratorbacterium hansenii could acquire antibacterial properties when combined with the right agent, right antibacterial agent, as I mentioned. So when we look at that, in an overall, so this multiple vegetable peel waste become effective not only to reduce the cost, but also recycle the kitchen waste effectively, as we mentioned in the beginning, with the huge amount of them. So in addition to the yield, high water holding capacity, porous structure, high moldability properties of the waste-based bacterial cellulose from the hansenii become very encouraging as a result of its characteristic, and it can be used also in the packaging and the delivery systems for that matter. Also, the physico-chemical characteristics show that bacterial cellulose may be produced from mixed vegetable waste, could also be applied as a suitable, promising for biomaterial for the medical and the pharmaceutical fields. And also, I just want to give you a very short ending for our works based on the waste valorization. It's a brand new, so we got a funding from the TÜBİTAK, so we're going to make a PHA production from the waste, which is a bioplastic, so from the bacterial strains, and using again the waste for that production. I'm a co-owner of a spin-off firm with the university also, and the back polyzyme bioengineering and the microbiota will make that 100-liter production from the waste, which is not seen in Turkey right now. Also, we also have different kind of waste valorization work, like the getting and acetic acid production using the cotton stalk as a feedstock, and also the getting caffeine and catechin from the black tea waste, which is very abundant again in Turkey, and also with the back polyzyme and the Marmara University, we had a patent application just using for the waste for the actually to have some biopolymers. So it is under the investigation right now. So this is my work. So I want to mention about the valorization of that different kind of feedstock into that value-added products. And thank you for listening. And I'm really appreciative and grateful for having me today. If you have any question, I will happily answer it.
Thank you very much for your nice and detailed presentation. If you have any questions, you can ask by turning your microphone on.
Benim aslında sorum yok. Soru da şimdilik gelmedi diye söze girdim. Hocam elinize sağlık. Çok güzel çalışmalar. Böyle biyomühendisliğin başından alıp sonuna kadar getirip gerçekten ürüne dönüştüren uzun soluklu çalışmalar olmuş. Elinize sağlık demek istiyorum. Ben keyifle dinledim.
Çok teşekkür ederim.
Çok teşekkür ederiz gelip bize vaktinizi ayırıp anlattınız. Elinize sağlık tekrar.
Çok sağ olun. Ben de çok mutlu oldum. Çok teşekkür ederim. Biraz geniş tutmaya çalıştım. Umarım sıkmamışımdır sizi. Yani farklı yerlerine dokunmaya çalıştım özellikle. O yüzden biraz kapsamlı olmuş olabilir. O yüzden kusuruma bakmayın.
Yani hocam evet, bu kadar çalışmayı tabii bir saatte yine de bize bayağı ben anladığımı düşünüyorum. Çok güzel
Çok memnun
aktardınız.
oldum. Çok
Dilinize
memnun oldum.
sağlık. Keyifle dinledim. Teşekkürler.
If no one has any questions, I would like to thank you again for your nice presentation.
Thank you very much. You're welcome.
And we can end this webinar right now.
Okay, thank you very much for having me here. So I really appreciate it. Thank you very much.
Çok teşekkürler.
Thank you very much.
Görüşmek üzere.
Hoşçakalın.